Hello.
Hello.
How are you doing?
I'm good. How are you?
I'm all right. So I know you know the drill. I feel like we've been on one of these test prep calls before where I
Yeah,
was the admin. I'm going to go ahead and give you host privileges, and
sure.
that way you have access to all the features that you need. And then I'm just going to unmute, or I'm going to mute, turn off the camera, and I'll just be doing my other work.
Yeah, sounds good.
You can just let me know if anything comes up at any point. I'm happy to hop back on.
Sure.
We'll just go ahead and give you host privileges, and then you won't hear from me for the rest of the session.
Yeah, sounds good.
All right.
Hello, everyone. Welcome to the mole cell bio session. I'm going to give it till 7:02, 7:03 in case people are rolling in. Okay, I'll get started. So today I'll be covering mole cell bio. Definitely not comprehensively. I don't think you could get everything in an hour. But I'm going to start with some kind of mole bio review, do a little bit of cell bio, and then I have like 2 practice passages that I got from Khan Academy, and then we can kind of work through those and answer. So it'll be kind of interactive in the second half. But at any point, feel free to ask any questions you have. As I'm talking, I might not see it in the chat right away, but like feel free to drop it in the chat or like raise your hand or something. I'll get to it eventually. So to start off with mole bio, the start of it all is DNA. In terms of the MCAT, I think it's important to know the structure of each of those 4. Like the amino acids, everyone says, but I think the DNA bases also come in handy. So know what the 4 of those look like, and then obviously how they bind. So thymine and adenine bind, and then cytosine and guanine bind. It's also important to know that the CG pair has 3 hydrogen bonds compared to AT only has 2, so CG is stronger. If you have 2 different DNA strands and you want to know like which one's bonded stronger, which one will be harder to break, you'll probably want to see how many CG pairs there are. Also, Chargaff's rules. Adenine and guanine are purines, so they have like 2 rings, whereas thymine and cytosine are pyrimidines, so they have 1 ring, and then AT bind, CG bind, as I said. And so also the sum of the purines will equal the sum of the pyrimidines, if you ever need to use that. Obviously, like the central dogma is kind of the main part of mole bio, and that's just the flow of information goes from DNA to RNA to protein. So DNA replicates itself to make more DNA, then the process of DNA going to RNA, transcription, and then RNA to protein is translation. And you're going to need to know all of those in detail, and any kind of questions about those could come up. So for DNA replication, there's a lot of different enzymes that come in play, so it's good to know what each one does loosely. So like helicase is kind of the one that unwinds the double helix and separates the parent strands at the origin of replication, so where the replication starts. And then topoisomerase, kind of like ahead of that, it kind of coils so that everything doesn't get knotted, so negative supercoiling to kind of reduce the strain from the unwinding. That kind of, if you're unwinding something that's bound together, you're kind of tightening it, so it kind of undoes that. There's single-stranded DNA binding protein, and so that binds to each strand to prevent like re-annealing. if it's a single-stranded DNA. Primase is what like synthesizes the RNA primers and puts them at the beginning of each DNA strand. You only need one for the leading strand, but on the lagging strand, you're going to have multiple for the Okazaki fragments. But I have a photo in like one of the coming slides. DNA polymerase is what actually makes the daughter strands in a 5' to 3' direction. So again, like the leading strand, it just goes like one strand, but the lagging strand is discontinuous. So it's just like different pieces and a lot of different primers. And then, yeah, DNA ligase joins those different fragments together. So this is what it looks like. So there's the parent strand that you have that unwinds, and the daughter strands are the two that you're making. And then these are the enzymes that work, and you can kind of see what I meant by the Okazaki fragments. So the leading strand is continuous 5' to 3, but then for the lagging strand, you have like different pieces going on. Helicase is what's unwinding, and you just keep going. So that's like the process of DNA replication. Videos, there's also like videos and stuff on YouTube that can visualize it nicely as well. And then from there, the next step is going to RNA. There's a lot of different types of RNA. So it's important to know they might like only put the abbreviation, so it's good to know what each one stands for. It can be kind of confusing. The main one is messenger RNA or mRNA. That's the one that's called coding RNA because that actually turns into proteins after translation. The rest are non-coding RNAs, and so some of those include ribosomal RNA. It helps, like it makes the ribosomes with proteins as well. There's transfer RNA, which pairs the codons with amino acids during translation. There's small nuclear RNA, which associates with different proteins to make like ribonucleoproteins. And those come into play with spliceosomes, which there's a photo of later. Small interfering RNAs or siRNAs are for RNA interference. And so that's like for degradation and like silencing. And then there's microRNAs also for RNA interference. They also both like bind complementary sequences to silence or degrade. So those two are kind of similar. So this is transcription by an RNA polymerase. You have your mRNA strand, your kind of sense coding strand, because that's going to be the one that's the same as the product you're making. And then the antisense is the non-coding strand. And so that's the one that's like complementary to what you're making. So that's how you're transcribing. And another thing is just it's going to be the same as the sense strand, except for RNA has uracil instead of thymine. There's different activators and repressors. These come into play a lot. Like you might have passages where you have something that's being activated or repressed, or something like changing in the genetics. So it's good to know different ways that transcription can be promoted or repressed. So transcription factors, you can see kind of like the activators on the binding site help like the promoter region, help transcription, whereas repressors can block transcription factors. And then splicing is the changes that happen to RNA. So as you know, there's like introns and exons. Exon, like the mnemonic is like exons are expressed. So EX, that's like the part that actually goes into your mRNA at the end, and that's what becomes the proteins. But there's a process to kind of get rid of the introns that are in between that. And so first you have your pre-mRNA, then they add a 5' cap, so just like some bases on the cap, and then a bunch of A's on the 3' end. And then you go through the splicing process. So introns are removed. Then you have your mature mRNA. Splicing, so as I mentioned, those other types of RNAs can come into a spliceosome, and that's what like cuts out the introns. And so the introns leave, your spliceosome is disconnected, and then you have just the exons. And then you move into translation. And so you have your ribosome there, the blue thing, and you have like the 3 different sites. So you first need a start codon, and that codes for methionine. You have your tRNA there that recognizes the codon in 3s, of course, and then you translate and you actually turn that into like adding a protein, and then it keeps going until the last one, and then you have a release factor that binds to any of the 3 different stop codons, and that's what stops translation. An open reading frame, or ORF, that's kind of like which kind of frame of threes you're reading in. If you're given a DNA sequence, you might have to, or an RNA sequence, you might have to figure that out. And so technically for any like strand, there's 3 different reading frames you could use, like starting from the first one, second one, or third one, and then any kind of combination of those. Start codon AUG is where you'd have to start from, and then there's different stop codons. So you can kind of look at like the different combinations or what happens when you're given a strand. This is also just another like illustration of the beginning and the end of translation. So as I said, the first like AUG, the start codon, it starts with the methionine amino acid. And so it'll be like scanning for that. And the termination at the end there, you have a release factor that recognizes your stop codon and enters the A site of the ribosome. And then it releases the tRNA and your peptide. So your peptide is complete and the complex disassembles. So that's how translation ends, and then you have your finished protein. And so this is the kind of larger grand scheme of transcription and translation. Important to note which parts happen where. For any process in general, it's good to know like where in the cell it happens. So our transcription and translation, that's happening. So transcription is happening in the nucleus, whereas translation is outside. So the transcription happens, and then those modifications like the poly-A-tail-5' cap happen, splicing happens, and then it's your mature mRNA that exits the nucleus. That's what you have that goes to the ribosome and gets translated outside of in the cytoplasm. So those like loose amino acids would be in the cytoplasm to make your proteins. After translation, your proteins, they can get processed. So the ribosomes are on the rough ER, so that's where your new proteins would be. And then they can go into vesicles, go to the Golgi apparatus that can modify it, and then be sent to where they need to go. So the Golgi could put on like a signal that lets it know where to go. That'll also be later in the cell bio part. Any questions so far? I guess now we're kind of moving into cell bio, so I'll stop for a minute if there's any questions. These slides are posted. Again, I think it's in the files section of the Canvas, but the passages at the end I have like the answers to, so I didn't put that in, but after the session I'll re-upload like the updated slides. So you'll have the passage with it as well. Okay, we'll move into the cell cycle. So different phases of the cell cycle, of course, we know mitosis is where the cell duplicates, but there's also interphase, so G1, S, and G2. G0 is if it goes into arrested cell growth, otherwise G1 is duplication of organelles, S is like synthesis, you duplicate the DNA, G2 you grow more, correct any errors, and then mitosis you divide. Cytokinesis is like the physical division, mitosis is like the chromosome separating. So that cycle goes on and on until cell death. In terms of cell division, there's mitosis in more detail here, and then also meiosis for gametes. So mitosis will just make 2 identical daughter cells, as we know, so it goes through prophase, chromosomes condense and stuff, and then the nuclear membrane disintegrates, so you just have your free chromosomes. Metaphase, they all line up. Anaphase, they separate. Telophase, you kind of start forming your 2 different cells, and cytokinesis, it pinches off. Meiosis, similar, but then there's like 2 of each of those steps, and they have like crossing over so that you actually like change up the DNA. So it's not identical daughter cells, you have 4 unique daughter cells, and that's for your gametes. So for meiosis 1, as you can see, it's like your chromosomes separate, but then it like in anaphase 1, and then meiosis 2 is where the sister chromatids separate. So that's the main difference there. Don't know if you can see my cursor. How much of meiosis and mitosis do you need to know? Yeah, that's a good question. I would say know what happens in each phase, the general process, like the difference between the two. It's obviously like kind of it might not come up on your test, it might come up. In general, for like if it comes up in a passage, they might give you like certain details if something's going wrong or whatever, so like you'll get that information, but it's still good to know what should generally happen in each phase. They might say like this problem happened, like what phase did it happen, and then like these are your 4 choices. So it's good to know the difference between like prophase versus metaphase, anaphase, what you can expect in each one, and of course the purpose of each process in general for the MCAT. Like when you're studying a process, know where it happens, like why it happens, overall like meaning of it, in case they ask anything about that. Another thing about meiosis that's good to know is if there's errors in meiosis. There can be errors in different ways. So non-disjunction, if it happens in meiosis 1, then it's like your homologous chromosomes, instead of splitting up evenly, 2 of them might go to one side and none to the other. If you do that, and then assuming meiosis 2 is fine, so the sister chromatids separate properly, you're supposed to have 23 chromosomes in humans, but if you have non-disjunction in meiosis 1, out of your 4 daughter cells, you'll have 24, 24, 22, 22. Sorry, guys. And then on the other hand, if you have the non-disjunction in meiosis 2, so your homologous chromosomes will separate properly, but then your sister chromatids both go to one side. Then half of your daughter cells will be okay, but then the other half you'll have a 24 and 22. So that's another thing that might come up, is if there's like an error in meiosis, what step did it happen in? So looking at the final results will show. So half of them will be okay if the error was in meiosis 2. Next thing, membranes are really important to know, I would say. The selective permeability of the membrane, only some substances can cross it with no energy. So like nonpolar molecules can cross the membrane. Water is polar, but it's small enough to cross. Large polar molecules will need a transport protein. There's also aquaporins, back to water, that can help increase the transport of water if you need. So in limited amounts, it can transport passively, but there are also other ways. Ions will need a transfer protein because they're charged. Then macromolecules cannot cross. And then you also want to know like the phospholipid structure with the tail and the head. So passive transport, again, that's when there's no energy required. And so you will go from high concentration to low concentration. Higher solute concentration creates higher osmotic pressure, which refers to the tendency of water to cross a semi-permeable membrane by osmosis. So this graphic, I think, shows the different types of passive transport pretty well as well. In terms of facilitated diffusion, it could be specific to just that molecule, or it could be non-specific in that anything can cross. For active transport, there's primary and secondary, as you can see there. And so for primary, the sodium-potassium pump is an example. And then you have like ATP hydrolysis that makes the concentration gradient for sodium. And then there's also secondary, where they're both going in the same direction. And so sodium is moving down its concentration gradient, and so then that helps glucose move against its concentration gradient. Then there's also, again, uniport, antiport, and symport. So uniport, one thing, antiport, 2 things in different directions, symport, 2 things in the same direction. The sodium-potassium pump is probably one of the most common like transport pumps that you should know. And so it's 3 sodium out, 2 potassium in. So that's important. You also have a phosphorylation step, also important to know. And then in the nerves, you'll have voltage-gated ion channels, also important to know. Maybe Angel covered this during the nervous system yesterday, not sure, but in terms of just action potentials, it's good to know where these play a role and how the calcium channel is closed. And then after depolarization, you have exchange of sodium and calcium and how that plays a role in how nerves work. Good to know. And then in terms of the cell, definitely not going to be comprehensive, but you should know in general like what the different organelles do, the organization of the cell. Again, kind of like it'll take up too much time if I get into everything today, but I chose a few organelles to talk about a little bit. Mitochondria, you're gonna have to know, powerhouse of the cell, of course, but know the structure of it beyond just that. So like the fact that there's the two membranes, the outer and the inner. The inner is folded into cristae, and that's important because that's where the electron transport chain is. And so that increased surface area with all the folds gives you more area for like the electron transport chain to happen, and that is like happening, the transport is across that membrane. You have your intermembrane space, and then inside the inner membrane is the mitochondrial matrix. All good to know. And like I said, you want to know like any process that you need to memorize, you should know where it happens. So like glycolysis is happening outside versus like electron transport chain, all of those things. Good to know where it happens relative to the structure. In terms of other organelles, this is just kind of like random ones that I put on, but lysosomes are kind of like the garbage bin of the cell. They have hydrolytic enzymes, so they can break down a lot of different things like waste products. Endosomes help transport, package, and sort cell material. The endoplasmic reticulum is kind of split up into rough and smooth. Rough is called that because it's studded with all the different ribosomes, and as we saw, that's where translation happens, and then those proteins get secreted into its lumen. The smooth ER is where lipid synthesis happens instead, so you can think of it as like protein synthesis versus lipid synthesis. It's also involved in the detoxification of like drugs or other like toxins. And then the Golgi apparatus is kind of like the post office of the cell. It like adds on different groups, so like post translation modifications that need to happen to proteins, and it also can put in signal sequences, and it directs where the proteins need to go. And if you've taken cell bio, I'm sure you know this in much more depth, but you know, whether it goes to the membrane, whether it just like floats around in the cytosol, that is determined by the sequences that get added. So that's the Golgi. And then I think the last thing I have is like the cytoskeleton. Provides like support and structure to the cell. You can see on the left there's kind of the three types of the fibers. There's microfilaments, microtubules, and intermediate filaments. Also good to know about those. Know like the subunits, so like actin for microfilaments or tubulin for microtubules. Intermediate filaments you might not need to know in as much detail, but the other two I would say definitely important. Know the difference kind of between the three, like what they help with and how motor proteins work. So for microfilaments, that's myosin, and then microtubules, there's kinesin and dynein. How those work and how things are kind of transported along those motor proteins. So yeah, that's also important kind of, you don't really think about it when you think organelles, but the cytoskeleton is very important as well to know. I'll pause if anyone has any questions at this point. Otherwise, we can get into the practice passages. Okay, I guess I'll get started on the passages. So I have two different passages. I guess the way I set up the slide, it's like the passage and then the question, and then like there's 6 questions for each passage. So what I'll do, I'll give you the most time on the first one before I scroll, I guess, and then I'll let you guys see all 6, and then we can kind of take it up, and if anyone wants to like share their thought process, we can all discuss. But I think in general with my practice as well, like it can be tempting to like question, see the answer, question, see the answer. But when they're all tied to a passage, sometimes seeing the answer of one kind of affects how you answer the rest. So in my practice, I preferred like per passage to at least finish all the questions and then look at the answers. So I guess we'll do it that way, but I'll give you the most time for this one because you actually have to read the passage here as well. But the passage will still be there for the rest of the questions, so you don't have to like take notes on it necessarily. And let me know if it's like too small to read. Hopefully not, but I had to fit it all onto the slide. I'll give the most time for this one, and then I'll start flipping the slides. I'll give like a 30-second warning, I guess, for answering this question. Okay. This is the second question for this. Feel free to, you guys could write down your answers, send me your answers, whatever you'd like, whatever you're comfortable with. If you're comfortable with sharing, feel free. Thirty-second warning. Okay, next question. $30. Okay, next question. Also at the end, if I rush some questions, we can go back. So don't worry too much if you missed one. Thirty seconds. Thirty seconds. Okay, last question. Thirty seconds. Okay, before I get into the answers, were there any questions that I didn't spend enough time on? Feel free to put in the chat. We can go back to questions. Or you can put a thumbs up if you are good to go to the answers. Second question. This one, right? We'll go over all the answers after, so don't worry about that. If you couldn't get any, it's totally fine. Are there any other questions anyone wants to like look at real quick before we go over? Or... Okay, if not, I guess we'll get into it just for the sake of time. If anyone would like to explain their answer, I'd love to hear from you guys instead of me just talking the whole time. So, does anyone want to explain? So, for one, it was, I guess, B. Does anyone want to explain? There's a graphic there as well, if that would be helpful. All right, if not, that's okay. I guess the graphic kind of shows us for bacterial DNA replication, it's actually bidirectional, so it happens both directions from the origin of replication. But as you can see, the origin of replication is where it starts there, and then at the end, you'll have your 2 new DNA strands. So it's leading to the production of 2 strands or molecules of DNA. So as replication kind of proceeds, you have the forks, and then they're moving in 2 opposite directions. And then at the end, you have 2 DNA molecules from the template DNA. Yeah. I guess that wasn't all the options. So a lot of these, like, you just want to see what's different about the different answer choices. So hopefully you know that, like, it's for DNA replication, so you'll know it's DNA, and then the question is just, is it 1 or 2? For question 2, it was this one. This one might be a little more complicated, so I guess I'll start explaining this one. Basically, so for DNA replication, the forward primer anneals to the 3' end of the bottom strand, like the antisense strand, and then the reverse primer anneals to the 3' end of the top strand, like the sense strand. And so DNA polymerase then, like, you know, it moves 5 to 3. So in the top right there, kind of just have like an example of primers. So you're looking for a reverse primer at the Sol site that can amplify YAC1. So the YAC1 coding sequence, it was given at the start of the passage, and they said, like, you know, like 976 random bases in between or whatever, but you're given that part of the sequence. And so for a forward primer, or for a reverse primer, sorry, you need the 5' to match that kind of end. So you need the reverse complement of the coding strand that you're given, but they also need to integrate the restriction enzyme site. So you have like the Sol site sequence and the also the YAC1. So basically, I think the graphic at the bottom kind of shows, but you would need to have the end of the coding sequence as well as the restriction site in your enzyme to be able to incorporate the 2. Hopefully that explanation makes sense. So it would be that option. Another kind of thing is just like you have to write it 5 to 3. So the way you might have thought of it was the 3' to 5' way, but you want to flip it back to get your answer. Any questions about that? I think that was, I think when I was doing it, the hardest question for me in the bunch. I don't know if, let me see the chat. I just realized the chat might not be popping up for me. Oh, the restriction site is like it's like a sequence of a few base pairs of DNA that bind to a restriction enzyme. So in this case, let me go back to the passage. Yeah, so the restriction sites, so the 2 restriction enzymes, those are the names, sorry, BAMHE and SOL are your 2 restriction enzymes. So the sites are where those 2 enzymes would bind, basically. And then you also have like the arrow of where they would cleave. Okay, if there's no other questions, move on to 3. The ampicillin, their gene, so that's to aid in the selection of successfully transformed antibiotic resistant colonies, not sensitive colonies. So we would assume the sensitive ones would die. So the ampicillin resistance gene, I think they put it in the passage mentioning it, but it's basically so it's included in the E. coli vector so that the bacteria expressing that, they will be resistant on the ampicillin plates. So then you can select for the vector of interest on the plates. So a lot of like lab techniques do come up in the MCAT in these like bio passages, so it's good to know. Okay, hopefully no more questions there. Also, if I move on and you still have a question, feel free to put it in the chat. The next question was about DNA ligase. If it was omitted during the preparation of the vector, would hybridization still occur between the ends of the YAC1 insert and the ends of the digested vector? So the answer is yes, but the bonding will be temporary because basically DNA ligase is for those phosphodiester bonds. So without ligase, like the base bearing between complementary bases can still happen, your hydrogen bonds, but they'll be temporary because you don't have like the stabilizing phosphodiester bonds that DNA ligase forms. So DNA ligase catalyzes the formation of those covalent bonds between adjacent DNA fragments, as you can see in the photo there, basically. So like the A and B coming together. All right, and then this is the 5th question, I think. Does anyone want to explain if anyone got colony 2? Okay, if not, no worries. So I pasted the table in just to, because that's kind of what it's talking about. But basically, if you cut with only one restriction enzyme, you'll get like one DNA strand, and the entire cloning vector is 3 kilobases long, kb long, including YAC1, and then the YAC1 is 1 kb. So if you cut with only one restriction enzyme, you'll get the single like 3 kilobase strand. If you cut with both, you'll get like the YAC1, which is 1 kilobase, and the rest, which is 2 kilobases, so 3 minus 1. And so that would be colony 2, because you have with just one. So like colony 4, the combination of the two worked out, but you're only getting 2 kilobases with BAM or salt only, which doesn't make sense. So it's colony 2 because of the 3-3, because the whole thing is, as you can see, like 3,000 or 3 kilobases long. But then you have like a 1 and 2 if you were to cut it up. So the combination of those. A lot of questions you'll see this pattern where it's kind of like the question you would just need to figure out like 2 different things, because like the combination of answer choices will just have like 2 different things that are different about them. So first you have to see, is it like, are you supposed to get 3-3 or 2-2 when it's like only 1? And then is it 3 when you have both, or 1 and 2 when you have both? So you can kind of split it up if that makes the information easier to digest. This last one, I guess it's more of a general knowledge question. DNA cloning and molecular biology research. You can use it for GMOs, you can use it to amplify genes and create recombinant proteins. But studying the process of like DNA replication in prokaryotes doesn't like directly involve DNA cloning itself. So I suppose that's just kind of, because DNA cloning is kind of creating identical copies of the particular gene of interest. So studying the process of DNA replication, you don't necessarily need cloning, was I guess the logic behind that one. Are there any lingering questions? I'm also pretty bad with monitoring the chat, so. Meiosis is in the M phase, yes. So in the, I guess, mitotic cycle, you do G1S, G2 mitosis, but in the meiotic cycle, you do G1S, G2 meiosis. And I think G2 might be a little different just leading into the meiosis part, but otherwise, yeah, similar. Okay, not seeing other questions, so we'll move on to the next one. So that was like what I would say a more mole bio type passage. This is what I would say is a more cell bio type passage. Hopefully there's enough time. I guess I'll do similar timings to before, but I'll give you the most time on this one so you can read the passage as well. Thirty seconds. Okay, next question. Thirty seconds. Next question. Thirty seconds. Okay, next question. This one I had to put the paragraph kind of small, so hopefully you guys can still read it. $30. 2nd last question. $30. And final question. $30. So for the sake of time, we'll move on. But also, I'll upload the slides that have the answers. So if you feel like you didn't get enough time, you want to work on it on your own and not be spoiled, you feel free to hop off right now. The answers are in the slides that'll be uploaded. So that's totally fine. I don't want to spoil the answers if you want to think about it for longer. But if you'd like to stick around, we'll quickly go over the answers right now. So for the first one, this is just kind of general knowledge about the lipid bilayer. As we said, ions cannot cross, and so oxygen is small and nonpolar, so that would be the one that can transport the easiest without a protein. In terms of this question, the least likely true is that the airway surface is basic. Basically from that paragraph that I screenshotted, it states how it's the CFTR protein doesn't operate by active transport, so it has to be passive, so that gets rid of choice D. There's other things that kind of counteract the other choices, buildup in chloride would increase the number of ions in the cell, so it would draw water into the cell. Choice B makes sense. So the other ones you can basically figure out are true. Another thing that just like also just proves that C is not true is that you have like the Cl minus and Hg3O minus. So bicarbonate is a base that flows out of the cell. So it makes the CFTR dysfunction in CF patients will then increase acidity rather than make it more basic, because that's malfunctioning basically. The next question here, amino acids, if there's like only one thing you memorize for the MCAT, it should be the amino acids. But anyways, there's a photo of them there. And you can kind of see a pattern with the answer choice there. You basically, it's a transmembrane domain. And so you'd want hydrophobic amino acids. And so the ones you have there, like leucine, methionine, valine, proline, like you have all like hydrophobic amino acids. So that's the most likely one. But there's a lot of questions like this where you just have to be able to like remember what group the amino acids belong to. For this question, it is that first graph. Let's see which graph. Relative chloride conductance. Okay, yeah. So from the table, like the class 3 to 6 mutations are like a decrease in ion transport or decrease in like the amount of CFTR protein. But class 1, 2, and 7 result in CFTR not being produced or not reaching the membrane. So you would have a like less chloride conductance with class 1, 2, and 7, and that's consistent with choice A. So I guess just about like reading that table. Then for this question, it was answer choice B. So you're deleting 3 nucleotides, so it's not a frame shift mutation. Wouldn't be a truncated protein. You're only losing 1 amino acid, and so what's the other one that impairs? Yeah, so basically improper protein folding would explain why like the misfolded protein is retained in the ER. And then is this the last question, I think? Yeah. Class 1, and that's the class that's caused by premature stop codon. So like they're generated by nonsense mutations. So when a mutation codes for a stop codon too early, and so it would be class 1. Are there any questions about the take up or about anything? If not, like I said, this will be uploaded as well, so you guys can go back. Passages were from Khan Academy, so you might see them if you're also doing the Khan Academy review. But yeah, I'll hang on. I'll stay on like a few more minutes for any questions if anyone wants to hang back. But otherwise, Angel is running a session tomorrow, and then I'm running one Friday. So hopefully I'll see you guys on Friday. Hopefully when I have a bit more of my voice back. But thank you all for coming.
All right. Hello. Thank you so much.
Thank you. Are there any, Lisa, do you have any questions? Okay. If not, then thank you.
Might have stepped away from their laptop.
Yeah.
But thank you so much for
probably.
your hard work and for the thorough session. I'll go ahead and end the call. And then you said that everything that you used today is going to be uploaded on Canvas
I'll upload
already.
it right now. Yeah.
Wonderful. Thank you
Bye.
very
Thank you.
All right. Have a good evening.